We're now going to discuss animal viruses. Animal viruses can be surrounded by a lipid protein envelope that is typically derived from the host cell. Because of this, it can include glycoproteins, and these glycoproteins can mediate binding to the host cell receptors. Most of these glycoproteins would have been made through the host cell endomembrane system. These lipid protein envelopes can resemble the host nuclear or the host plasma membrane. This gives an idea of the mode of infection. Some infections will be resultant in the cytosol, whereas other infections will be localized to the nuclear area. These animal viruses are classified according to the type of genome. This can be RNA or DNA-based viruses. This genome will serve as a template for transcription or translation and replication. There are many diverse and creative methods of reproduction and infection within the classes of animal viruses. This diagram gives an overview of the 6 different classes involved in the classification of animal viruses. Note that there can be multiple different ways to classify animal viruses. Some classification systems have 7 to 8 groupings. However, for the purposes of this course, we are referring to the 6 classes as seen here. The first class of animal viruses includes double-stranded DNA viruses. These can include your adenoviruses, herpes virus, papillomavirus, etc. Some of these viruses will have an envelope, such as herpes virus, whereas others do not, such as the adenovirus. The second class of viruses refers to single-stranded DNA-based viruses, such as the parvovirus. The third grouping refers to double-stranded RNA-based viruses. The fourth group refers to single-stranded RNA viruses. Note that when a single-stranded RNA virus is injected into the cell, it can immediately serve as an mRNA molecule for translation. The fifth class of viruses includes single-stranded RNA viruses as well. However, in this case, these viruses would be serving as a template for mRNA synthesis. This means that the single-stranded RNA in the virus would have to be modified in order to create the mRNA for translation. Finally, the sixth class of viruses includes single-stranded RNA viruses once again. However, in this case, the single-stranded RNA serves as a template for DNA synthesis. This DNA sequence would then be injected into the host cell genome. This is common for retroviruses, such as HIV. This diagram here gives you an overview of the various types of viruses and their classification. Note that on the left-hand side of this diagram, we can see that there are both DNA viruses as well as RNA viruses. Within that classification, you can have viruses that are double-stranded DNA viruses. They can be enveloped, meaning enclosed in a capsule, or they can be non-enveloped. Within the RNA viruses, we can have both single-stranded and double-stranded RNA viruses. As seen in the DNA viruses, some of these will be enveloped and others will be non-enveloped. This slide here gives an overview of the 7-stage classification system for viruses. Note that some are going to be double-stranded DNA or single-stranded DNA, as well as single and double-stranded RNA. Some classes of RNA will be positive and negative. However, when you have a positive RNA molecule, it will have to be converted to a negative RNA, which will then be used as a template for mRNA. Note that the overall goal of all of these classification groupings for viruses is to create an mRNA molecule. This mRNA molecule will then allow for the host cell to produce more viral particles. This slide here gives an overview of the different types of infections that are common in humans. Note that some viruses are completely specific to a tissue type. However, other viruses have adapted to affect multiple tissues within an organism. This diagram here outlines the three modes of infection for three types of viruses. First, we have a double-stranded RNA virus. Note that once the viral genome is injected into the cell, the positive RNA will be used as a template. This can be directly translated to make viral proteins. It can also be used as a template to create the negative strand of RNA, thus recreating the genome of the virus. These will then be assembled and then be able to leave the cell to further infect adjacent cells. The second part of this diagram is the mode of infection in a positive RNA virus. This is also considered to be single-stranded. Once the strand of RNA enters into the host cell, it can be translated into different types of proteins for the virus. We can also see that replication proteins will make a negative RNA strand. This can be used as a template to make further positive RNA strands. These positive RNA strands will be assembled with the virion proteins to once again create an infected virus. This can then leave to infect other cells. Finally, in this diagram, we have a typical retrovirus. In this case, once the host is infected, a reverse transcriptase enzyme will turn the RNA strand into a DNA fragment that can then be injected into the host genome. Once this is injected into the host genome, it can easily be transcribed and translated. This will then lead to the assemblage of more viral particles. Note that many of the viral particles will assemble in the host cell membrane. Thus, as the retrovirus RNA leaves the cell, it will be encoded with the host cell protein. Once again, this will increase its ability to infect a host cell. A good example of a retrovirus is HIV, or the human immunodeficiency virus. A retrovirus as a class is a virus with a single-stranded RNA genome that can engage in reverse transcription. When this genome, that is single-stranded RNA, enters into the host cell, the enzyme reverse transcriptase that is usually present in the virus itself will copy out the ssRNA and make it into a DNA fragment. This viral DNA can then be permanently incorporated into the host cell genome, at which point we would call this genetic sequence the provirus. The provirus can serve as a template for further mRNA production that could eventually lead to the synthesis of viral proteins, or it can also be used to make more copies of the genome to assemble new viruses. Proviruses can lay dormant within the host genome for many years, or it can sporadically become active. HIV specifically targets the T cells of the immune system. This is why we call it an immunodeficiency disease. Thus, when the virus attacks these cells of the immune system over prolonged periods of time, it can lead to the development of AIDS, which is acquired immunodeficiency syndrome. This is also one of the reasons why HIV outbreaks in an individual will commonly occur when their immune system is compromised, either by medication, by general health, or through the infection of other diseases. This diagram here gives you the overview of the life cycle of the HIV virus. Note that the HIV virus has the presence of the reverse transcriptase enzyme within the genome. It also is encoded with a glycoprotein coat in the viral envelope. Here we can see that once the virus attaches to the host cell, the genome will be and reverse transcriptase will be entered into the host cell cytoplasm. The reverse transcriptase will act on the viral genome to convert it into an RNA-DNA hybrid. This will then be replaced by a full DNA molecule. Once that occurs, it can now be incorporated into the host genome where it forms a provirus. This provirus will serve as a template for more mRNA molecules as well as a template for protein synthesis. We can then see that new viral particles can be easily made within the host cell. They will periodically leave the host cell and then be able to affect adjacent cells. There are many defenses that we can have against viruses. One of those defenses is to ensure that we have a healthy immune system and overall health. Prior to exposure to a virus can lead to immunity in some instances, or it can also lead to a better recognition of the virus in order to be able to fight it faster. Good general health can also minimize some of the viral effects and decrease the infection period, thus limiting the spread of the disease. Vaccines also offer protection against viruses. Vaccines typically have pathogen derivatives that are used to expose the host immune system to the virus. This will then allow for the organism to be able to remember what the virus is within its immune system. This can sometimes lead to lifelong immunity for certain diseases, or it may lead to an increased ability to fight the disease once an organism has been exposed to it. There are also many antiviral medications that target specific viral properties. These can be used to prevent, minimize, or eliminate the infection. It is also important to note that antibiotic treatments will have no effect on viral diseases. This is because antibiotic treatments are specific to bacterial properties. As a result, they do not work against viruses. Some viruses can be termed as emerging viruses. We use this term to describe viruses that suddenly become apparent within a population. This can be used to describe HIV and AIDS and its appearance in the 80s, West Nile disease and its appearance in the early 2000s, Ebola in two appearances, one in the 1970s as well as a more recent outbreak in 2014, Zika virus that we've recently encountered, as well as COVID-19. An epidemic refers to a widespread outbreak of a disease. However, an epidemic is usually localized to one or a few locations, whereas a pandemic refers to a complete global outbreak of a disease. It is important to note that many of these diseases, though they are termed emerging viruses, have been pre-existent within a population. However, they do not cause widespread disease, usually until a mutation arises that allows them to increase their rate of infection. It is important to know that RNA-based viruses have much higher rates of mutation than, say, their DNA-based counterparts. This is because there is no proofreading ability in RNA polymerase, which allows for more mutations to accumulate very quickly. These mutations would essentially negate any previous immunity that the host cell would have had. This can also lead to the evolution of more virulent strains of the disease. Some viruses can remain hidden in a population until a mutation occurs that increases their infective rate. This would then lead to an outbreak, at which point the disease would be noted. Some viruses have animal reservoirs. In this instance, the animal population would act as a reservoir for the disease. Many times, within the animal reservoir, the animal will not experience any adverse symptoms. This allows for the virus to easily maintain itself within these populations. Many times there can be a mutation that would then allow for human infection. At this point, the virus would be transmitted from animals to humans. Most of these mutations can be due to genetic recombination with other existing viruses that already exist within the host organism. That additional virus may have the ability to become infective for humans. Through genetic recombination, that advantage of being able to infect humans would then be passed on to the virus that previously did not have that ability. At this point, this could lead to a disease outbreak. Prions are considered to be protein-based infectious agents. They are not considered to be viruses. These prions are primarily due to a misfolded version of a protein. Once the misfolded version of a protein is inside of a host cell, it can cause the misfolding of properly formed proteins. These can then at times form aggregates that can disrupt cellular function. Prions typically have a long incubation period, usually more than 10 years, and they're also considered to be much more stable than the normally folded protein. Many of these are heat resistant, which means that with cooking they would still not be destroyed. This is why these Prions can be rapidly spread through cannibalism or through the consumption of infected tissues. We have seen this in the case of scrapie, which infects sheep, mad cow disease, which infects cows, however, can also affect humans if they consume the tissue, as well as Cura disease. This now brings us to the last topic of this course, one that we have all been dealing with over the last few months, COVID-19, also known as SARS-CoV-2. This is termed a novel coronavirus because it is a new type of virus within the coronavirus family. It is considered to be a positive single-stranded RNA-based virus. The average size is about 0.125 microns. Take a look at the diagram to the right to see the overall layout of the structure of this virus. The RNA is enclosed in a protein coat. This is then surrounded by a lipid membrane, and that lipid membrane has many spike proteins attached to it. These spike proteins are very important to allow for easy entry into the host cell. What is unique about this disease is that these viruses can replicate in both lung and respiratory tissues. Most coronaviruses can replicate either in one or the other. The current origin of this disease is currently unknown. However, by examining the genome, we have been able to learn some important information. 96% of the genome is similar to a previously identified Yunmin bat virus. This is strong evidence to support that this virus did indeed originate from the bat population. However, one of the main important differences is that the coronavirus now has a receptor binding domain on the spikes that can facilitate human infection. This would be an adaptation for human transmission that we do not see in the Yunmin bat virus. However, we have seen this domain pop up in other coronaviruses, such as the pangolin coronavirus. Originally, it was proposed that this disease originated from either bats or pangolins in the Wuhan market. However, when looking at the pangolin coronavirus, the genome is only 90% similar. This once again indicates that there's a higher similarity and higher probability that this did originate from bats. Understanding the evolution and structure of this virus is key, both for understanding where this virus originated from and also to understand the best way to combat this disease. And with that, you have reached the end of Bio 103. I hope you have enjoyed the course.